Yeah. The summer program in neuroscience, excellence and success, better known as SPINES. Can I hear SPINES in the house? Here we are. SPINES is a course that was established by Jim Townsend and Joe Martinez over 30 years ago. So we'll talk about that a little bit. Thank you for coming out to the Cornelia Clapp Auditorium and to those of you who are joining us virtually tonight. Throughout this summer, we are recognizing past members of the MBL community who have made extraordinary contributions to this storied institution. It is my privilege to share with you tonight highlights of the life and work of Jim Townsend and Joe Martinez Jr., who have been lifelong friends and collaborators, and faculty members, investigators, and corporation members here at the MBL. They first met, in fact, here at the MBL in the Frontiers in Teaching and Research program in 1974. Together, they went on to establish many programs that were aimed at providing professional development, scientific skills, mentoring, and networking, and have influenced the lives and careers of hundreds of individuals, including the 408 now alumni of the SPINES course. As of this year, we've broken 400 students, so that's wonderful. But they have also influenced the lives of many more than that in many other programs that they have established over the years, especially for underrepresented groups in the sciences. Joe Martinez, featured over there, was born in Albuquerque, New Mexico in 1944, and he remained proud of his Chicano heritage his entire life. He began his faculty career in 1972 at Cal State University, San Bernardino, in the Department of Psychology. He recognized that the health and well-being of Mexican Americans was neglected in the field of psychology, so he helped to organize the first symposium on Chicano psychology. And then he co-edited the book called Chicano Psychology, which established the field and remains influential after more than 40 years. Later in his career, he helped found the National Hispanic Science Network, which is dedicated still to improving the health equity of Hispanics. Joe Martinez served on the faculty of several institutions, including 25 years at the University of Texas, San Antonio, where he founded the UTSA Neuroscience Institute. And that institute crossed disciplinary boundaries. His early neuroscience work demonstrated the impact that drugs and neurotransmitters both have on learning and memory. And his later work examined the neurochemical, electrophysiological, and molecular substrates of synaptic plasticity. In addition to publishing more than 100 peer-reviewed papers in neuroscience, Martinez co-edited several books on the neurobiology of learning, memory, and sleep. Over the years, he was successful in obtaining nearly $18 million in funding to support both research and education. Throughout his career, Martinez was a passionate advocate for individuals from underrepresented groups, and he trained dozens of undergraduates, graduate students, and postdoctoral fellows in his own lab. Jim Townsend was born in 1935 in Albemarle, North Carolina, and he grew up in Harrisburg, Pennsylvania. He majored in biology at Virginia State University and participated in the ROTC and graduated with high honors. Because he himself couldn't afford to go to medical school, he began his career in the US Army. While giving lectures on biology for the Medical Service Corps, he became fascinated by basic science, and he decided to retire from the military. He earned his PhD in physiology at Purdue University and was recruited immediately to the faculty of Virginia State. Later, he accepted his postdoctoral traineeship at Harvard Medical School in Ed Kravitz's laboratory, which galvanized his passion for neuroscience. He also served on the faculty at Meharry Medical College and the University of Illinois in Chicago before he retired in 2010. When he arrived at Meharry, Townsend quickly obtained research grants from the National Institutes of Health and the National Science Foundation, and he built a strong research program that really made significant contributions to the trafficking of proteins that are involved in neurotransmission. And he created a culture that benefited students. He hired 2 active neuroscientists to the faculty and secured competitive federal funding in support of research. He created a multidisciplinary graduate program that earned an NIH training grant and developed an NIH-funded collaborative program with Vanderbilt University for predoctoral trainees. Jim Townsend also had an outstanding track record of training. Of 11 Black trainees, 9 remained in research and are now faculty members at research universities. Together, the 2 of them, Jim Townsend and Joe Martinez, collaborated with the Diversity Program in Neuroscience, a nationwide training program for pre- and postdoctoral scholars. This program offered 3 important components in addition to the training stipend. It offered mentorship, annual monitoring, and enrichment activities. The enrichment program included a month-long experience here at the MBL, encompassing professional development, lectures in neuroscience, mentoring, and networking. It became the summer program in neuroscience, ethics, and survival, which is now known as the summer program in neuroscience excellence and success, or SPINES. It was officially launched as a course at MBL in 2000, but SPINES has continued to maintain an outstanding track record of success, with many of its alumni now in senior academic positions. This Joe L. Martinez Jr. and James G. Townsend endowed distinguished lectureship, which supports a special lecture during the SPINES course, was established in 2006 to recognize their extraordinary work and to prepare the next generation of neuroscientists. Both Jim and Joe sadly passed away last year in 2020, and their memories were honored by generations of grateful colleagues and mentees. Tonight, we're pleased to honor Joe Martinez and Jim Townsend and their enormous contributions to advancing neuroscience discovery, to increasing diversity, equity, and inclusion in the neurosciences, and to the Marine Biological Laboratory. Thank you for joining me tonight in honoring their legacy. Thank you. And now I'd like to turn it over to Ellen Lumpkin, a professor in the Department of Molecular and Cell Biology at the University of California, Berkeley, and co-director of the MBL Neurobiology course, who will introduce tonight's speaker. Thank you.
Good evening. It is a thrill to introduce our lecturer tonight. In fact, I can't think of a more appropriate honoree to give this lecture. Our lecturer is Dr. Diana Bautista, who is the 1943 Memorial Chair and Professor of Cell and Developmental Biology and of Neurobiology at UC Berkeley. I met Diana for the first time more than 20 years ago. We were both just starting to work on somatosensation, so we were budding sensory neuroscientists. Diana was a graduate student at Stanford University in Rich Lewis's lab, and I was just finishing my postdoc and starting my own lab at UCSF. Diana, for her postdoc, did some remarkably heroic recordings, electrophysiological recordings of tiny, tiny currents that are important for activating the immune system. And she was looking around to see what might be an interesting topic for her postdoc. She came to my poster and we hit it off right away. By serendipity, Diana also moved to UCSF within a year or two, and both of us have been collaborating on and off ever since. A hallmark of Diana's research is her creative approach to research and her fearlessness to tackle high-risk, high-reward projects. As you'll see tonight, her scientific interests appear to be remarkably broad, but there is a unifying theme, and it's really quite simple. She uses bold, multidisciplinary approaches to solve scientific problems that have the potential to relieve human suffering. For her scientific contributions, Diana has been recognized by numerous awards, including a Rita Allen Scholar Award, the Society for Neuroscience Young Investigator Award, a Howard Hughes Medical Institute Scholar Award, and recently an NIH Director's Transformative Research Award. In addition to these contributions, Diana's career is notable because of her sustained commitment to educating students from traditionally disenfranchised identities in the STEM fields, including students here at MBL. She's been a faculty member in the neurobiology course since 2009, so this is her 12th summer, and she currently serves as co-director along with me and Mark Hammerland. I've had the pleasure of co-teaching with Diana on principles of physiology and neuroscience for several years now. The clarity and excitement with which she presents mechanisms in environmental sensing are simply inspirational. As you're about to experience, she is a terrific teacher. Please join me in welcoming Diana to the stage.
Okay, let's see, lots of technical difficulties with this microphone and my earrings. And now let's see how Zoom's going to handle this. Okay, share screen. The sharing, okay, looks good. And now, let's see if I share the right screen. Yeah, I know. Okay, great. Thank you, Ellen, for that really nice introduction. And thanks to everyone who's here today and on Zoom for joining me this evening. It's really an honor to give this lecture for a number of reasons, especially tonight, where we get to honor the legacies of Drs. Martinez and Townsend. Though I never met them, I benefited from their work in many different ways. And if I'm not sharing.
We'll get this.
I'm going to say exactly what I just said.
Yeah.
Now.
You.
Oh, we practiced this twice and it worked really well. Okay, great. Okay, so as I was saying, I've definitely benefited from all of the amazing work that Drs. Martinez and Townsend did, both indirectly and directly, as I was an American Psychological Association Diversity Program in Neuroscience awardee. And so definitely benefited. Dr. Martinez was the director of this program for several decades and benefited from both financial and intellectual contributions from being part of this amazing group. I'd like to say that their work has really done a lot to diversify neuroscience, but that we also have a long ways to go. There's still a lot of disparities, especially looking at Black, Latinx, and Indigenous students, where persistence in science is far less than expected from their representation in the general population, and also compared to other groups who declare STEM early on as a major, with disparities at both the bachelor level as well as the PhD level. And there's also disparities at the professoriate. So currently at degree granting institutions, 33% of professors are women, far lower than the general population and those who start out in science. 7% of professors are first in their family to attend college, 5% come from low-income families and are Pell Grant recipients, and only 6% come from underrepresented groups. And as someone who checks off all of these categories, I'm really grateful for programs and the outreach of many people, including Dr. Martinez and Townsend, for providing a sense of belonging, for promoting diversity, equity, and inclusion in science. I'm really grateful and want to take their work and move it forward. I'd also like to thank. Could also say that I used funds from this fellowship that I had to attend my very first scientific meeting here at the NBL when I was a second-year graduate student. And it was in this very same auditorium that I attended my first scientific talk at a meeting. And it was delivered by Nobel laureate Erwin Neer. And it was very inspirational. And so now I feel a little nervous being on the same stage, but I'll do my best. And let's see, is this going to advance? Oh, I didn't do my little check boxes. There we go. So I'm here to tell you about our science, and I'm really excited to tell you about the work of an amazing group of scientists and trainees that I get to work with as part of the Sensory and Epithelial Neuroscience Lab at UC Berkeley that I co-direct with Professor Ellen Lumpkin. And we are interested in understanding how you feel. And while we do care about your emotional state, we're talking about the sensations, how you experience and feel sensations of itch, touch, and pain. Aristotle was the first to define the 5 senses that allow us to perceive the outside world. And while we know a lot about the molecular mechanisms and cellular mechanisms and circuit mechanisms of hearing and vision and taste and smell, our sense of touch is still a mystery. Compared to all of these other senses, we know the least about it. And to me, that's really surprising given how important touch is in our everyday lives. We know that touch is important for emotional well-being. The bond between a parent and a child is first forged through touch. Premature babies in incubators have regular scheduled periods of tactile interaction with caregivers and family members that allows proper brain development. So we know touch is important for not only emotional well-being, but brain development and normal development. And also, I think all of us over the past 18 months have really felt the impact that the loss of physical contact with our friends and loved ones, the consequences of that loss on our emotional well-being. Now, our sense of touch is also important for everyday tasks that we often take for granted. So touch-sensitive neurons mediate our ability to walk in a straight line without staring at our feet, to tie our shoes, maybe the thing that we do the most these days, texting, and also more complex behaviors, learning to read Braille or the ability to learn to play piano are mediated by touch-sensitive neurons. And altered tactile sensitivity is the #1 complaint in the clinic. Some examples include tingling and numbing sensations that come with repetitive use injuries like carpal tunnel syndrome. Diabetic neuropathy can lead to a loss of tactile sensation that triggers repetitive injuries that are the ultimate cause of amputation associated with diabetes. Chronic itch is a debilitating condition that's on the rise in many countries, and I'll talk about it a bit later. And also chronic pain associated with tissue injury or diseases like cancer can be very debilitating. And while we consider pain as an irritation, acute pain at least as an irritation, it can also play an important protective system. So pain plays an important role in allowing us to avoid noxious encounters in our environment. So it's a warning system. If you're cooking and you grab a hot frying pan, you have a reflex that allows you to drop that frying pan before you cause too much tissue damage. There's also a memory component or a learning component to pain. Hopefully the second time you go to cook, you remember that you should grab a pot holder so you don't burn yourself. You learn to avoid things in your environment because you want to avoid that pain signal. Even inflammatory pain serves a protective function. So a burn, a sunburn, an injury, your sprained ankle, as some of you in the class know, allows you to have time to heal, right? So that inflammatory response is there to help clear injured dead cells, damaged cells. The inflammatory mediators trigger some swelling, but also allow healing and encourage you to protect that injured site and to also maybe next time cover up or be a little careful. But chronic pain, that is defined as debilitating pain that lasts for 6 months or more, no longer serves any purpose. And it can occur after an injury where these abnormal pain sensitivities never go away, where your system never resets back to baseline. Serves no purpose, and it's very difficult to treat. And to highlight the importance and both the importance and the bane of the pain system, there's a couple of articles that I'll point out if you're interested. So this is Ashlyn Blocker, who suffers from congenital inability to detect pain. As a baby, she was constantly injuring herself, was constantly self-mutilating, but never cried or expressed any expression of pain or discomfort. Her parents recall times when she was cooking pasta with the family and dropped the fork in the boiling water and reached in and grabbed the spoon and had no sensation of that experience at all. But from learning and from nagging, supportive nagging from her parents, knew maybe I shouldn't have done that. And this has led to multiple injuries, sprained ankles. She was even playing basketball once and broke her leg and didn't know and continued playing even though her bone was exposed. And so lack of pain, we don't want to treat pain and we don't want pain to go away because we know that it's very important. And so these types of syndromes highlight the importance of this warning system. But at the other end of the spectrum, constant pain syndromes, this is Cameron Bosdog, who suffers from complex regional pain syndrome, where she feels incredible burning pain sensations in her extremities without any stimulation at all. And so I think this highlights the yin and yang of the pain system, where we need to be able to dampen the pain system in the case of Cameron, but also keep a normal pain system to allow healthy activities and have a healthy nervous system in the case of Ashlyn Blocker. Chronic pain affects more than 10 million Americans annually. And right now there are no treatments, there are no cures, and even the drugs that are out there only partially alleviate the debilitating pain that these sufferers experience. And that is really the challenge. How do we deal with this? What we want to do is to discover genes that underlie chronic pain so that we could develop new therapies that target pain at its source. So I'll tell you a little bit about our somatosensory system that mediates our sense of itch, touch, and pain. This is just a schematic of the human body where our somatosensory system, which is just groups of neurons shown here in green, diffusely localize and innervate the entire body. They innervate the skin throughout the body as well as the viscera. These sensory neurons detect a wide variety of environmental stimuli, including innocuous and noxious pressure, temperature, and also a variety of chemicals, both from the outside world. If you've gotten lemon juice in your eye, your somatosensory system mediates that. And also these neurons monitor internal state and are very responsive to a variety of endogenous chemicals in the body. And in my lab, we not only study a variety of primary human neurons and other human cells that contribute to sensations of itch, touch, and pain, but we also use the mouse as a model organism. This is a whole mount embryo of a mouse where all of the somatosensory neurons are beautifully labeled with green fluorescent protein so you could see them. And there's a lot of conservation between the mouse and the human nervous system. And here are the cell bodies of the dorsal root ganglia neurons that mediate somatosensation from the neck down. And here are the trigeminal ganglia cells that project the free nerve endings and innervate the head and the neck and the oral cavity, where they mediate not only sensations of itch, touch, and pain, but also things like food quality, texture preferences in food as well, something that Professor Lumpkin's very interested in. So the somatosensory system is not just one type of cell, but there are many, many different subtypes of neurons that mediate different types of environmental stimuli and mediate signaling from the outside world in. And roughly, though, they come in two flavors. There are a subset of distinct subsets of free nerve endings that terminate as free nerve endings that mediate itch and pain. And these are often referred to as nociceptors because they mediate nocifensive responses to irritants, such as that may cause itch or pain. And the other, say 50% of the somatosensory system is composed of a diverse set of mechanoreceptors or touch-sensitive neurons that mediate proprioception responses to gentle touch. And we are interested in how these cells take environmental stimuli and convert them into electrical signals, which is the currency of the nervous system. And I'll just walk you through that. So we know that somatosensory neurons detect environmental stimuli in four main steps. The first step is here, it's transduction. It's conversion of a physical stimulus into an electrical signal. And so temperature gets converted to an electrical signal through a variety of specialized proteins. Same thing with mechanical force. And we're interested in defining transduction molecules. Signal transmission is the electrical signal that travels from the periphery to the spinal cord, the first relay in the central nervous system. And you need the signal to make it all the way there for you to start to initiate the signal transmission to the higher order levels. There's a lot of modulation of the signals that enter the spinal cord. And we also know that then the signal travels to the brain for you to experience perception. So pain is an emotional experience that can be triggered by the transduction of a noxious stimuli. So pain is in the brain, and transduction we often refer to as nociception, the initial conversion of a noxious stimuli into an electrical signal. But pain is in the brain. So one of our goals is to identify new pain targets in the periphery that mediate transduction. So how do we go about doing this? And we are addressing some key questions in pain biology to help understand this. The first question that we want to understand is, how do innocuous and noxious stimuli excite sensory neurons? So what is it? What are the molecular force transducers or thermosensors that do that? We also want to understand how pain thresholds are set so that we could distinguish between the brush of a feather or the prick of a pin. And we want to understand how injury or disease leads to altered pain sensitivity, such that somebody who suffers from chronic pain feels the brush of a feather as incredibly painful, and someone who suffers from diabetic neuropathy might step on a tack and not feel it at all. And the other switch that we're very interested in is between itch and pain. So under conditions of chronic itch, pain feels pleasurable. So how is it? How do these different disease states trigger these switches in how we perceive different types of stimuli? And what I'll do for the rest of the time today is just tell you about 3 different approaches that we've been taking to start to understand how our somatosensory system functions to gain insight into these different processes. So the first approach is to use natural plant products that target somatosensory neurons. There's a long history of using natural products that target the somatosensory system to understand the pain system. And I just want to highlight some of the perhaps most popular ones that you've all experienced. Capsaicin is the pungent compound in chili peppers. So if you felt heat when you eat chili peppers, it's through capsaicin, the active component. menthol, the pungent cooling compound in plants of the mint family, and the brassica family produces pungent isothiocyanates that trigger a bit of an inflammatory response. For example, when you eat sushi with wasabi. And by studying how these different plant compounds trigger these different psychophysical sensations, this led to the identification of several protein sensors that play a key role in somatosensation. So TRPV1 is a capsaicin and heat-sensitive ion channel. So in response to heat or binding the active compound in chili peppers, this protein in the plasma membrane of neurons opens, allows electrical signals to come in, and that triggers that electrical impulse that's required for that signal to go from the periphery to the brain. TRPM8 is a cold and menthol sensitive ion channel, and TRPA1 is the wasabi receptor. And I spent a lot of time characterizing these two channels, and I'll just tell you a little bit about them, focusing on TRPM8. So TRPM8 gene was cloned. What's remarkable about this gene is that you could express it in any type of cell and confer both cold sensitivity and menthol sensitivity onto that cell. And so here's just an experiment where cells are in a dish, they're expressing the TRPM8 gene, and that protein is functional. And instead of looking at electrical signals, we're using calcium imaging as a proxy for neuronal activation. Purple means that the cells are in a quiescent, non-excitable state. And in using a pseudocolor scale here for this fluorescent dye, activation is shown in red. And what I'll show you in this experiment is the application first of menthol, followed by cold stimulus. If I find my mouse, oh, here it is. Okay, so first you'll see menthol. First was a control. Here's the menthol. You see this is a menthol sensitive cell. And then we applied a cold stimulus and the cell increased in its activity. So the same cells that are menthol sensitive are also sensitive to cold. We could also measure electrical currents directly across the cell membrane using electrophysiology. And when we did this and applied a range of temperatures, what we found was that TRPM8 encodes a ion channel that is temperature sensitive. At body temperature, it's not active, so there's no current. But as we decrease the temperature, we find that you could see an increase in activity as the current flows into the cell and has a set point right about 10 degrees Celsius, which is the same temperature at which human psychophysical studies show that people start to experience cold pain. And this was really exciting because this told us that TRPM8 might be a mediator of cold pain in vivo. And to test this directly, we generated a TRPM8 deficient mouse. And I'll show you one behavioral assay. We've done many to ask if TRPM8 is required at the organismal level for cold sensitivity and cold pain hypersensitivity associated with disease or injury. So we designed a temperature preference chamber where we could control the floor plate temperature and set them to any temperatures that we wish. In the experiment I'll show you, we have a plate that's clamped at 30 degrees C. Mice like that a lot. And the other side is not really noxious, but it's 20 degrees. And we can just ask, where does the mouse prefer to stay? And where's my, okay, so first I'm going to show you a mouse that is wild type. So it has a normal copy of TRPM8. Here's 30 degrees, here's 20 degrees. And you don't have to have a PhD to see that this mouse doesn't really like the 20 degree side. It has a clear preference and it spends all of its time on the right. Okay, here's its mouse's brother. This mouse lacks the TRPM8 gene. So And here. You could already see a difference. Even without quantification, this mouse no longer has a preference for the 30/20°C. And we could do this for a range of temperatures. This mouse will stand, and we didn't let it, but it will spend a lot of time on a freezing cold 0 degrees plate without any sign of discomfort. And we went on to do a variety of other experiments looking at different types of disease states and specifically cold allodynia, and found that trypamate plays a key role in a variety of cold-evoked hypersensitivity phenotypes in mice. And also studies looking at human patients have linked mutations in trypamate to a severe form of trigeminal neuralgia that's incredibly debilitating and painful, as well as genetic variants associated with chronic migraine and cold allodynia, where cooling, normally innocuous cool, feels incredibly painful. And also, trypamate is a key target for treating pain associated with chemotherapy-induced peripheral neuropathy. About 60% of patients who are treated with chemotherapy develop peripheral neuropathy. And of those patients who develop it, about 40% of them go on to have neuropathy for the rest of their life, even though the cancer may be in remission and the chemotherapy has stopped. This symptoms include tingling or pins and needles sensation, paining, burning, or numbness, and severe sensitivity to hot or cold, and cold allodynia is a hallmark. And there's currently clinical trials ongoing right now looking at trypamate antagonists to see if it's effective in blocking cold allodynia associated with CIPN. So the other approach that we use is to study animals with unique tactile systems to gain insight into how our nervous system can detect mechanical force, gentle, innocuous touch, and also painful stimuli. And one of the animals that we've been very interested in studying is the Starnose Mole, and using this as a model for somatosensory mechanotransduction function. This is a very interesting animal. We collaborated to work on this animal with Ken Catania and also Duncan Leach, who's in the spines course right now, and he was a graduate student with Ken. This is a really fascinating animal that has the most sensitive tactile organ in the animal kingdom. It uses this star organ, these appendages that you see here surrounding the nose, as a way to hunt for food and small insects that other animals in the very competitive ecosystem of the tunnels and in the wetland areas around rivers, that they used as more like a hand to probe the surface looking for small insects that other animals can't detect. And this is a great movie that I have to, hopefully can show you. Where Ken Catania took this animal, both the Starnose mole and the Eastern American mole, put it on a glass plate, dissected very small pieces of worm that graduate students can't detect at all, even with our very sensitive fingertips, and asked how does a specialized Starnose mole versus a non-specialized mole that doesn't have a star organ fare in this with this array. So here's the movie with the Eastern American mole. You can see that this mole comes, it licks up a couple of these worms, but basically walks right over them and does not eat them. And here's the Starnose mole. So it's really great at detecting very small insects and does this remarkable behavior where these longer appendages reach out and touch a food source. These smaller appendages surrounding the mouth then touch an object a second time, and only then do you see the little tongue come out and lick the object. So this is not only the most sensitive tactile organ, this is the world's, this animal has the Guinness Book of Records award for fastest mammalian forager. And it's thought that in something like the blink of an eye, it touches something twice and eats. So it's remarkably sensitive and remarkably fast. And three very talented trainees in my lab, Kristen Gerhold, Takeshi Morita, who's teaching in neurobiology, and Rose Hill, decided to take a look at the touch-sensitive neurons that innervate this tactile organ to probe their function. What types of touch-evoked responses do we see in the neurons? And to also, look to see the molecules that are enriched in the tactile sensitive neurons that innervate the star organ. And by doing this, they identified a variety of molecules that we think play a key role in touch, not only in the Starnose mole, but also that are expressed in the nervous system in other mammals, including humans and mice. So again, we took some of these candidates and knocked them out in mice to ask, do they play a role in normal somatosensation in non-specialized animals? And one of the molecules that we identified, called S1PR3, we found is a regulator of mechanical pain and pain hypersensitivity in mice. So if we take a knockout mice versus a heterozygous or a wild type litter mate, what we found in response to light touch, the animals were completely normal. They had a normal response. But when we looked at painful stimuli like pinprick or applying a heavier force that triggers a paw withdrawal reflex in wild type animals 100% of the time, that many of the animals didn't respond at all. And those that did only responded about 40% of the time. So they had a very diminished pain, mechanical pain sensitivity. And through a variety of experiments using knockout animals, as well as some commercially available S1PR3 inhibitors that were designed for other reasons but didn't work out, we found that it's a key regulator of touch. So if we decrease its activity, we found that that triggers mechanical hyposensitivity, such that mice become insensitive to the pinprick. Under normal conditions, it plays a very important role in setting normal mechanical pain thresholds. And in response to elevation of its agonist or elevating activity of this S1PR3 receptor, we found that it can trigger chronic pain and mechanical and thermal hypersensitivity. And we're now very interested in looking at the role of this key molecule in a variety of different disease conditions, including neuropathic pain and inflammatory pain models. And the third approach that I'd like to tell you about is our recent work probing the mechanisms of inflammatory disease. And we've been very interested first in studying mechanisms of itch and inflammatory and chronic itch, and most recently atopic dermatitis. So acute itch at least is defined as an irritating sensation that causes a desire to scratch. And I'm sure you've all experienced the itch of a mosquito bite or an allergic rash or a brush with poison ivy, which I know some of our faculty have had running this week. And perhaps you've come up with creative ways to scratch your itch. And itch, like pain, plays an important protective role. And so maybe we don't think about it so often, it's just a nuisance, but itch can be protective, both keeping us away from a variety of skin burrowing insects and biting insects that transmit a variety of diseases. And so I know that mosquitoes really like me and I get really itchy, and so I really avoid them. I'm really good at hearing that buzz and covering up or putting on some DEET before they come near me. Scratching is thought perhaps to trigger the removal of burrowing insects before they could lay eggs or transmit diseases and spread. But itch is not also, since I'm at the marine biology labs here, itch is not just for hairy mammals. It's thought that itch is highly conserved. And so fish have come up with creative ways to scratch that itch or to get rid of parasites that they no longer want by rubbing against coral, or in the case of this eel here, going to a cleaning station where other fish are encouraged to come and remove parasites. And soon as those parasites are removed, then the eel will snap and go away. So we think that it's highly conserved. And it's thought that even Drosophila have a version of itch. They have a very directed behavior to mite infection, and that's very different from a painful response or a tactile grooming response, where there's a very directed removal of a mite that's crawling across its back. But chronic itch is very different. So if you can imagine the very worst itch that you ever experienced, and imagine what your life would be like if that itch were to spread over all the extended parts of your body, and what life would be like if that itch were present for every second of every day. And that's what people who suffer from chronic itch describe. Very debilitating, and it's very difficult to treat. So 1 in 10 people at some point in their life will suffer from chronic itch. Perhaps allergies or skin conditions like atopic dermatitis or eczema come to mind. A variety of systemic conditions like kidney or liver disease can trigger chronic itch, some cancers, infectious diseases, and there's a growing list of drugs that have chronic itch as a side effect. And it's thought that different mechanisms associated with different diseases trigger the chronic itch, but what they have in common is that antihistamines are not effective. So there's a lot of interest in identifying new targets to treat chronic itch with the idea that there will be new drugs and therapies available. And if you are interested, there's a very intriguing article in the New Yorker called The Itch that describes a patient who had nerve damage associated itch, and the drive to scratch was all consuming, such that she actually scratched through her skull and gave herself brain damage, as you could see in the CT scan. It's a really incredible article that really talks about how all-consuming neuropathic itch can be. So we spent the last 10 years and many other labs defining the molecules and cellular players in acute itch signaling, and that got us very interested in understanding chronic itch. So we set out to understand the interaction between the epithelial cells or keratinocytes that play a key role in itch pathology, the immune system, and the sensory neurons, and how this trio of cells interact to drive a disease state. And we decided to focus on atopic dermatitis or eczema, the most common chronic itch disorder. It affects up to 20% of children and about 10% of adults worldwide. And it's characterized by a very itchy, red, flaky skin with chronic itch as the main complaint. We were also very interested in looking at the role of the immune system and the nervous system in driving atopic dermatitis or eczema because it's an entryway to other diseases. So about 70% of patients who have severe atopic dermatitis go on to develop food allergies, allergic rhinitis, and a severe form of asthma that's difficult to treat in something called the atopic march. And so we thought if we could gain some insight into the mechanisms of atopic dermatitis, this might give us some ideas about how this atopic march progresses. Now, atopic dermatitis was primarily considered a disease of the epithelium and the immune system, and there wasn't that much when we first started looking at this to really try to understand how the nervous system fit in. And so, but we knew that you had to activate the nervous system at a minimum to get itch sensations to go from the periphery to the central nervous system. And so Rose Hill and Carolyn Walsh, 2 graduate students in the lab, decided to probe the neuroimmune interactions that drive the development of atopic dermatitis, as well as a transition from an acute to a chronic disease state. And what they found is they identified a number of key ion channels and receptors in the epidermis, including the RI1 channel that is responsible for calcium-dependent release of inflammatory mediators in atopic skin. And we found that sensory neurons play a key role in setting up the initial inflammatory response, and that the TRPA1 ion channel that I told you earlier is a wasabi receptor, plays a key role and is activated under disease conditions to trigger the release of inflammatory mediators that can then regulate the vasculature and promote the influx of immune cells into sites of inflammation, including neutrophils that we showed play a key early role in mouse models of atopic dermatitis. And we also showed that sensory neuron immune cell interactions play a key role in the transition from acute to chronic itch, and surprisingly showed that sensory neurons express a variety of traditional so-called immune cell receptors that were thought to exclusively act in the immune system. And instead, we found that there were a variety of key cytokines that act directly on the nervous system to, again, promote neurogenic inflammation and trigger itch and inflammation. And that they played a key role in this traditional type 2 immune response associated with atopic dermatitis skin. Now, interestingly and surprisingly, we found that these peripheral neuroimmune interactions in the skin triggered a wave of inflammation that traveled from the periphery to the central nervous system. We saw dramatic changes in excitability genes and upregulation of markers of inflammation at the level of the dorsal root ganglia and also in the dorsal spinal cord, suggesting that these peripheral neuroimmune interactions trigger plasticity, astrogliosis, and also influx of microglia. And if we block these peripheral neuroimmune interactions, we block this entire wave of inflammation. And what our lab is doing now is trying to define the neuroimmune interactions in airway inflammation and what triggers this atopic march from the skin to the spinal cord to the lungs. We think that these peripheral neuroimmune interactions are playing a key role in this transition. This work has also spurred our interest in understanding the role of the nervous system. We know that in the airways. So we know that the lungs and the upper airways are densely innervated by a variety of sensory neurons that express many of the same receptors and channels that we identified in atopic dermatitis. And that the trigeminal nerve, the vagus nerve, and the dorsal root ganglia all densely innervate the lung and airways and play a key role in homeostasis and inflammation. And these neurons play key roles in acute sensory responses, sneezing and coughing in response to irritants. And we want to understand how these peripheral neuroimmune interactions drive the switch from homeostasis to airway inflammation in the context of asthma, and also in the context of SARS-CoV-2 infection and COVID-19. And this is the new area of research in my lab. And we're really excited to start to look at this. And we also know that these sensory neurons that we've been studying, the trigeminal ganglia that innervate the upper airways, have been shown to be directly infected by SARS-CoV-2, as well as in addition to the airway epithelial cells. So we're trying to understand how SARS-CoV-2 infection of airway epithelia leads to the release of inflammatory mediators that are associated not only with atopic disease, but found as part of the cytokine storm in patient biopsies. We want to know the consequences of SARS-CoV-2 infection of sensory neurons, and how does that impact the recruitment of immune cells, the release of inflammatory neuropeptides that we know can trigger inflammatory responses. We want to look at changes in neuronal excitability. And also there's been a lot of controversy about whether or not there is or is not viral infection of the central nervous system, but these infected trigeminal ganglia may be a key sentinel for transmission from the periphery to the CNS, because the trigeminal ganglia innervate the brainstem and reside in close proximity of the neurons that regulate the key respiratory center. So these are some of the key questions that we'll be pursuing over the next few years. And finally, I hope overall that I've conveyed to you that our basic science approach has led to this discovery of novel therapeutics for treating human diseases. We love basic science. We are really curiosity driven, but a really nice side effect of our research is that we do gain insight into some, we think, very important disorders of the nervous system that hopefully other scientists and pharmaceutical companies might be interested in developing novel therapeutics or drugs. And with that, I want to thank all of the wonderful people who did the hard work in the lab. I think I named most of them during my talk. I'm really fortunate to have wonderful collaborators and trainees who do the hard work. Mentors have played a key role in how I approach science, so I have to thank them as well. And I want to thank my funding agencies who gave me the money to do this work. I feel so lucky about that. And I also want to thank the MBL for the invitation to give this talk tonight and for allowing me to be a part of the neurobiology course, which has really made my summers really wonderful, has pushed my science in different directions, and has allowed me to meet amazing scientists who I look forward to working with every year. Thank you.
Thank you, Diana. And we didn't have a 4th of July parade this year, but we'll come back strong next year and you'll be here for it, I know.
2019.
So we have time for just a few questions, both here in the auditorium and online. So please just raise your hand and one of us, Donna or I, will come to you.
Hey, thank you for the talk. That was wonderful. I was just wondering if you could elaborate a little bit more on how something that we consider as like a peripheral sensory channel, like tryptophan, might be affecting something that we consider to be more of a central nervous system disease, like chronic migraine.
Yeah, so it's a great question. So what we see in a lot of pain syndromes and chronic itch is that there might be a lot of activity that starts in the peripheral neuron, but that activity and hyperactivity or prolonged activity under conditions of chronic pain or chronic itch then lead to a wave of central changes at the level of the spinal cord. And we know that there's a lot of plasticity and changes in the cellular circuit at the spinal cord level that then drives changes at higher levels as well. So we think that there's sort of a wave that things might start in the periphery, but that pain, chronic syndromes then get solidified at the level of the spinal cord to create long-term changes in the absence of any persistent stimuli.
All right, hello. Yeah, so there appears to be an increasing body of evidence supporting that pain and itch at the level of the spinal cord are very intertwined and perhaps oppose each other via many mechanisms. And I'm wondering if you could comment on whether that oppositional relationship exists at the primary afferent level, or if perhaps there are more parallels between pain and itch at the primary afferent level.
Yeah, there are so many interesting interactions between itch and pain. So I'm sure you've all scratched an itch. If you scratch to the same degree a non-itchy part of your skin, it doesn't feel good, it hurts. And so what is it? There's that interaction at that level. It's known that there's a spinal circuit whereby peripheral pain neurons block signals from the itch pathway from being transmitted to higher levels. So there's definitely a block at the level at the spinal cord. And then there's more complex higher level influences and circuits that with top-down modulation that also can trigger a reward. I see people scratching, it's okay. Talking about itch, it's normal if you're really itchy right now. And so there's a lot of, the itch scratch cycle and the interaction between itch and pain, I think, is really fascinating. And people are starting to map out those central circuits now, but there's a lot to learn. And I think the question is, if we mask pain, there's some evidence that you trigger itch. So some people who are receiving morphine develop really incredible itch. And oftentimes then, at least I've heard in the clinic, would prefer to have the pain and stop taking the morphine. And so Dr. Lerner can speak to this, who's our itch expert in the audience. Anyway,
I'm not going to speak to that, but,
okay.
but this was a, you covered a fabulous range, and I happen to be sitting next to my daughter, formerly a non-STEM person, who noted that she understood every single thing you said.
Great,
So
that
that's,
was the goal.
But so my question is, what is a free nerve ending? And where does it end? And what's it in touch with?
What is a free nerve ending? I guess a free nerve ending is defined as a peripheral nerve ending that does not wrap around a non-neuronal cell or other structure. So the beautiful neurons that Ellen studies wrap around synapse with Merkel cells, wrap around hair follicles, are associated with Pacinian corpuscles, and the nerve endings don't interact or wrap around anything, but they are deeply embedded in and surrounded by skin epithelial cells, keratinocytes in the epidermis. And so they're not really free, but and we think that they're really important because not only do they transmit signals about itch or pain to the nervous system and fire action potentials, but when they're activated, they also release a lot of inflammatory peptides at sites of inflammation and regulate the vasculature. And so we are very interested in them in the context of skin inflammation, but also airway inflammation and in the gut under different disease states.
So we have time for just a couple more. I think we have at least one or two online. So let's go for one online to start.
Is the S1P reference mentioned in your talk associated with sphingosine 1 phosphate?
Yes, it is. So the sphingosine 1-phosphate receptor 3 is the molecule we found that was very enriched in the Starnose mole. And you can't, the Starnose moles don't breed in captivity, so you can't make knockouts. So in our mouse model, what we found that S1PR3 is a very important regulator of mechanical pain under normal conditions, but that under inflammatory conditions, high levels of S1P, the ligand for this receptor, triggers hyperactivity, and that can lead to a different phenotype, which is pain hypersensitivity. So this same receptor seems to be playing a baseline sort of tonic role in setting mechanical thresholds, but when it's overactivated by having high levels of this endogenous inflammatory mediator, then you start to see pain hypersensitivity kick in.
Wonderful talk. Really enjoyed it. I wanted to talk a little bit about CREPS again, that chronic regional pain syndrome. I knew a young woman, she's a middle schooler who was very active and a good sportswoman, and then developed CREPS to the point where years later she had to have a limb amputated in order to try and take care of it. So you talked about peripheral inflammation. Does that have anything to do with it? And what's the best treatment right now? Do you know for CREPS and what can?
Yeah, it's a great question. There are a lot of pain syndromes where there are very few effective treatments, and CREPS is one of them. In many cases, especially children, there's some trigger, there's some injury done, that occurs during playing sports or something that triggers it, and then the system is primed and basically never resets. There have been some therapies that have been effective for some people, but they're not pharmacological, so there are not really any good drugs to treat it, but there were more different types of therapy in sort of training the immune system or the nervous system and trying to change the way you do activity. So for example, some people exercise who have CREPS and then become hot. And so thermoregulate to not trigger an event. And so it's more behavioral therapy that I think have been really effective, but it's something that many people are interested in understanding.
Do the mechanisms you study also show how we become numb to pain?
How we become numb.
Numb to pain.
Oh yeah, I'm very interested in numbing and tingling sensations and what that means. Is it the absence of pain? Is it activating something else? And I think there's evidence for both. One of the natural products that we've been interested and have been studying include the active compounds from Szechuan peppers that are not spicy hot, but give you a tingling, buzzing sensation, like putting a 9-volt battery terminal on your tongue. I see those who laugh have done it before. And that sensation turns out is a combination of dampening nociceptors, so silencing a subset of nociceptors by blocking some sodium channels, but also activating a really interesting bursting activity in a subset of large diameter mechanoreceptors. So the frequency of firing of these bursts that you get it's very similar to the buzzing sensation that you feel from eating these peppers. And there's been some other interesting experiments, like putting a blood pressure cuff on and then releasing it, causing that pins and needles sensation. And so, but the molecular mechanisms, I'd say, are still up in the air.
Great. Let's give Dr. Bautista a hearty thank you.